for the next two sessions. We do have a sponsor for this session, Azero Solutions. Azero works in partnerships with a whole bunch of different groups to protect the richness of our water bodies by offering design and installation services for watercraft decontamination stations. They do have an exhibitor booth that you can check out through the lobby. So go check them out. You can ask them questions and see what they're all about. With that, I'm going to pass it over to our moderator for this session, Sydney. Sydney is our Aquatic Invasive Species Coordinator, so she is well equipped to take on the role as moderator for this session. And I will pass it over to you, Sid. Take it away. Enjoy.
Thanks, Mac. Hi, everyone. I'm sure you're as excited as I am for the session today. Before we begin, just a quick housekeeping item. I ask that you please submit your questions for speakers using the Q&A feature. And then I'll read the questions for the speaker at the end of each presentation. So to kick us off, we have Kate Schwartz and Emma Ehrenfeld from the St. Lawrence River Institute. Kate and Emma, over to you.
Thanks, Sydney. I'm just going to share our screen real quick here. All right, and we'll go from the beginning. All right, so hello everyone, and thank you for joining us for our talk about our observations over 10 years of nearshore monitoring on the upper St. Lawrence River. We'll be going over some results from a recent paper on the expansion of the invasive tube-nosed goby as well. But before all that, we'd like to just quickly introduce ourselves. My name is Emma Ehrenfeld. I'm a research assistant on the FINS project at the St. Lawrence River Institute, and my favorite fish is the bowfin.
My name is Kate Schwartz. I'm also a research assistant on the FINS project here at the River Institute. I have a degree in science from Bishop's University, and my favorite Ontario fish is the small but mighty tadpole mad tom.
All right, so just some background. The results and observations we'll be sharing today have largely been generated by a nearshore fish project called FINS. The FINS project was started in 2015 as a joint effort between the River Institute and the MCA's Environment Program to address community concerns and general knowledge gaps about small fish in the upper St. Lawrence River. Community members had been seeing shifts in the health of game fish and bait fish populations, which were largely attributed to industrial contamination in the area. And at the same time, researchers were noticing a lack of data on the distribution and health of small minnow species like the pug-nosed shiner, and wondered what interactions and connections these species might have within the fish community as a whole. A nearshore fish project like this would also provide insights into the distribution of introduced and invasive species in the area and the impact they might have on local fish communities. So since 2015, the crew has collected a wealth of data on the nearshore fish communities and habitats of the upper St. Lawrence River. With over 600 surveys and more than 230 sites from Prince Edward County to Montreal, we have observed a wide range of habitat types and community compositions. This year was a milestone for the project, not only because it is our 10th year, but also because we caught our 200,000th fish. It is certainly an impressive number, as the total of 67 species we have encountered over many survey sites. The FINS project has completed many nearshore surveys in the upper St. Lawrence, right from the very edge of Lake Ontario, even up into the Ottawa River and some smaller tributaries. While the Cornwall and Akwesasne areas are very well documented now, as you can see by the density of the points, we've also expanded the scope of the project to cover some lesser documented areas such as Morrisburg, Cardinal, and Maitland. We have also been able to visit some critical habitat in areas such as the Thousand Islands and confirmed that these areas continue to be refuges for sensitive species despite the changing conditions and introduction of invasive species like the zebra mussel and round goby. We'll continue to monitor year-to-year changes to gain valuable insight into the impacts of long-term changes such as climate change and how local species are adapting. As you can see from the previous slides, we have been very lucky to monitor a large section of the upper St. Lawrence River over the years. The data we've collected informs monitoring efforts throughout the St. Lawrence River and is shared with our partners, including the Department of Fisheries and Oceans and the MFFP in Quebec. The longevity of this project has allowed us to track many invasive species throughout the section of the river, and we have even been around to see the rise of some newcomers to the nearshore scene. But first, we'll talk about the well-known and well-documented invasive species, the round goby. When we first got started with this project, round gobies were already very well established in the area, and this fish was and continues to be one of the most abundant and common species we encounter at our sites, and it easily makes the top 5 every year. And while many of us know to look for the characteristic black spot on the first dorsal, we have found a few gobies that lack this spot, and there's some interesting work being done to investigate those spotless gobies. So as we mentioned, the round gobies are one of the most abundant, often most caught species at our sites, and this map shows that they appear at nearly every site that we visit in the FINS project. The next species is one that you might not be quite as familiar with. The Restrian tube-nosed goby was introduced around the same time as the round goby, but their populations have not expanded as rapidly until more recently. Although found in Lake St. Clair in the 1990s, they were not observed in St. Lawrence until 2016, and their habits don't seem to be as aggressive or detrimental compared to the round goby. However, we do not fully understand how they might be affecting the nearshore communities just yet. And this map shows all the sites where we have encountered tube-nosed gobies during our nearshore surveys with fins. They have moved downstream considerably and are now being found in Quebec waters as well. 2024 saw a huge jump in tube-nosed goby abundance, going from a total of 442 individuals in 2023 to over 2,240 individuals last year. One site alone had over 330 individuals. So here's a simple graph just showing the percent occurrence of invasive gobies over the years. Round gobies were already very abundant in the river when the FINS project began, so their line, which is the blue one, starts off quite high on the graph. We found them at a minimum of half of our sites every year, but it is usually much higher, and we're typically finding round gobies at upwards of 70% of our sites, and this has been especially high in the recent years. Meanwhile, tube-nosed gobies did not appear in our surveys until 2018, where they were only seen at 4 sites and in very low abundance. Since then, we've seen a pretty rapid increase, both in abundance and percentage of sites they're showing up at. In 2024, tube-nosed gobies appeared at 76% of our sites, which was a huge jump in only a few years.
So since we're already talking about tube-nosed gobies now, we wanted to talk to you guys a little bit about results from a recent paper. So as you may have seen from the previous slides, extensive work has been done to analyze the data collected from sampling efforts of tube-nosed goby. And recently, some of our colleagues completed a paper on the density and expansion of the species in the upper St. Lawrence River. While this paper is still in press, like I mentioned, we still wanted to share some results, so you guys get a sneak peek of that today. The tube-nosed goby was initially undetected in the upper St. Lawrence River between 2004 and 2015. They were first documented near the Thousand Islands in 2016, with their population density significantly increasing thereafter. The tube-nosed goby's distribution in the upper St. Lawrence River extends from Kingston, Ontario to St. Zotique, Quebec, having expanded 225 kilometers downstream from 2016 to 2023. You can see on the maps to the right, hopefully I know they're a little bit small, that the yellow points representing tube-nosed goby captures drift downstream. The average distance for annual downstream expansion was around 32 kilometers per year between 2016 and 2023. The highest densities of tube-nosed gobies were detected upstream between Kingston and Morrisburg, Ontario, with a hotspot around the Thousand Islands where they are initially detected. Tube-nosed goby density gradually declined downstream, reaching their lowest densities in Lake St. Francis near us here in Cornwall and Valleyfield, Quebec, hinting at a possible invasion front in this area. Our findings revealed a significant effect of seasonality on tube-nosed goby density, with an obvious peak around mid-July. That's when we tend to find young of deer fish and adult fish. On this slide too, I just wanted to briefly give a shout out to John Farrell at Thousand Islands Biological Station and get Jessica Goldschmidt from the Government of Quebec for their contributions to this project. We couldn't have done it without you. Our results support a growing presence of tube-nosed gobies in the nearshore fish community. In 2021, the tube-nosed goby was a minor component of the fish community, but increased significantly in 2022. In 2023, the last year of this study, the goby was reaching densities comparable to those of the round goby. We also found a positive correlation between round goby and tube-nosed goby densities, indicating that these invasive species tend to occur at the same sites. It's very interesting that they do so because they tend to have different habitat preferences, with tube-nosed gobies preferring more heavily vegetated areas, but both of these species are just so abundant now that there's significant overlap. Tube-nosed goby body size varied significantly with both time of year and location in the upper St. Lawrence River. The total length of gobies increased during the summer days to reach a maximum body length in late fall, though occasionally large adults were also found in the summer. Larger tube-nosed gobies were detected both in proximity of their original invasion site near the Thousand Islands and at the forefront of their expansion into Lake St. Francis, forming a sort of sideways hourglass size distribution, if you can picture that. Smaller individuals were found between Cornwall and Brockville, and at times were quite abundant, as you can see by that photo on the bottom there. Those are hundreds of young of the year fish that we encountered near Cornwall. Now on to a fish that is a little bit more uncommon in our surveys, although quite abundant near Montreal and Saint-Jean-de-Richelieu, and with small populations in Lake St. Francis, we simply do not catch many tench while we're seining. This is likely due to the species' preference for heavily vegetated areas, and these are very difficult to sample using the seine, of course, for anybody who's tried, and other traditional methods as well. They also seem to be sensitive to vibrations and disturbances in the water, which doesn't help our case. Some organizations have had success with electrofishing and also the use of environmental DNA when targeting this species, so we decided to give that a try as well. We are able to analyze water samples from our 2021 field season to compare detections using eDNA and seining. So here you can see that we've only observed tench at a couple of sites, Ganawage and also Lake of Two Mountains near the Ilotorc Bridge. Over the years, we have not found very many of them. While we have returned to these sites, we do not consistently catch tench year to year. Some years we do not see them at all. The majority of the tench we do catch are young of the year fish, while larger adults must be too quick or inhabiting deeper water that are not accessible with the seine. To account for these gaps, we are taking water samples to be analyzed for eDNA. Using eDNA, we detected tench at 48 sites compared to 1 observation made with seining in 2021. For these hard-to-catch species, and I won't say rare because we know that they're there, this combination of seining and eDNA seems to be successful in maximizing our chances of detection. Now on to an invertebrate. While not the focus of our nearshore work, we do often make observations other than fish while visiting our sampling sites. One such species is the rusty crayfish. Our observations of this species have been limited to the Cornwall area, thank goodness, but most of them occur around Akwesasne and the surrounding islands. Although our team has only seen a couple of these crayfish, their presence in Akwesasne is cause for concern. This area contains an abundance of important and untouched wetland habitat that is home to sensitive species like bridal shiner and grass pickerel. The introduction of this aggressive invasive species could have effects on our native wetland species. Our partners at the Mohawk Council of Akwesasne's Environment Program will be conducting benthic invertebrate sampling in the next coming years as part of their nearshore sampling to continue to monitor these sites. So a couple of species that we luckily have not seen is the marbled and red swamp crayfish. Both have been present a little bit further west, with the marbled crayfish being seen in Burlington. This species, of course, is asexual and so represents a significant threat if it does break from its isolated pond, and the red swamp crayfish is very aggressive. So we will continue to monitor for these species, although it will likely be quite a while until we see them. Plants as well. So as part of our nearshore work, we conduct standardized aquatic vegetation surveys at each site. A total of 8 to 10 meter squared quadrats are positioned throughout our sampling site, and a percent cover of each species is monitored along shore and also out to a depth of approximately 1 meter. We use this plant data to try and determine preferred habitat for species at risk like the pugnose shiner, but it has the added benefit of helping us document invasive species. So as you can see here, this is a map depicting the richness of invasive aquatic plant species at our sites from 2015 to 2024. The maximum number of species found at each site is 4, with all of these kind of hotspot sites being near Montreal or within wetland type sites with slow-moving water. The only significant change in terms of which species are present or not that we have observed is the growing presence of azolla or mosquito fern that was first observed in Ganawage River, where they are very well established. However, we have since seen it further downstream, such as in Long Sault in a small bay on Hoople Island, And the species is a cause for concern since it floats and is easily spread by downstream currents and wind, driving it into stagnant bays where it might take root, not literally, of course. And we will continue to monitor our sites for these species as well. Luckily, we have not seen any water soldier or European water chestnut, but again, we will continue to monitor our sites for these as well. And with that, I'd like to thank you guys so much for listening to our talk, and you can feel free to reach out to us with our emails that are listed there.
Thank you so much, Kate and Emma, for that presentation. That was great. We do have a question in the chat. What is known or suspected about small fish species introductions? Aquarium trade, ballast.
So in this area, there were a couple of species that we didn't mention, such as goldfish, which of course are very prevalent in the aquarium trade. We haven't seen many of those aquarium species present in our surveys. The marbled crayfish, again, is one that is common in the aquarium trade. But luckily, those observations seem to be limited to more urban centers such as Montreal and Toronto. Luckily for us, we do not see very many of them here. And if they are here, they're not very well established.
Lucky indeed. That's the only question we have in the chat, but folks, if you think of other ones, they both shared their emails on their slide, and then also in the chat, the Invasive Species Center just posted a Google Doc that will have the emails for all the speakers there. So thank you so much, Kate and Emma. That was great.